Hello, welcome to Biology, the scientific study of life. The DNA replication machine as a stationary complex. It is traditional and convenient to represent DNA polymers molecules as locomotives moving along a DNA railroad track. But such a model is inaccurate in two important ways. First, the various proteins that participate in DNA replication actually form a single large complex. A DNA replication machine, many protein-protein interactions facilitate the efficiency of this machine. For example, helix works much more rapidly when it is in contact with the primes. Second, the DNA replication machine is probably stationary during the replication process. In eukaryotic cells, multiple copies of the machine, perhaps grouped into factors, may encode to the nuclear matrix, a framework of fibers extending through the interior of the nucleus. Recent studies support a model in which DNA polymers molecules reel in the parental DNA and extraordinarily made daughter DNA molecules. Additional evidence suggests that the lagging strand is looping through the complex, so that when a DNA polymers complex synthesis of an Okazaki fragment and dissociates, it doesn't have far to travel to reach the primer of the next fragments near the replication fork. This slapping of the lagging strand enables more Okazaki fragments to be synthesized in less time. Proofreading and repairing DNA. We cannot attribute the accuracy of DNA replication solely to the specificity of base pairing, although errors in the completed DNA molecule amount of only 1 in 10 billion nucleotides. Initial pairing errors between incoming nucleotides and those in the template strand are 100,000 times more common, an error rate of 1 in 100,000 base pairs during DNA replication. DNA polymers proofread each nucleotide against its template as soon as it is added to the growing strand. Upon finding an incorrectly paired nucleotide, the polymers removes the nucleotide and then resumes synthesis. The action is similar to fixing a typing error by using the delete key and then entering the correct letter. Mismatched nucleotides sometimes evade proofreading by a DNA polymers or arise after DNA synthesis is completed by damage to an existing nucleotide base. For instance, in mismatch repair, cells use special enzymes to fix incorrectly paired nucleotides. Researchers spotlighted the importance of such enzymes when they found that a hereditary defect in one of them is associated with a form of colon cancer. Apparently, this defect allows cancer-causing errors to accumulate in the DNA at a faster rate than normal. Maintenance of the genetic information encoded in DNA requires frequent repair of various kinds of damage to existing DNA. DNA molecules are constantly subjected to potential harmful chemical and physical agents, reactive chemicals in the environment, and occurring natural in cells. Radioactive emissions, X-rays, and ultraviolet light can change nucleotides in ways that can affect encoded genetic information, usually adversely. In addition, DNA bases often undergo spontaneous chemical changes under normal cellular conditions. However, changes in DNA are usually corrected before they become perpetuating mutations. Each cell continuously monitors and repairs its genetic material. Because repair of damaged DNA is so important to the survival of an organism, it is no surprise that many different DNA repair enzymes have evolved. Almost 100 are known in E. coli, and about 130 have been identified so far in humans. Most mechanisms for repairing DNA damage take advantage of the base pair structure of DNA. Usually, a segment of the strand containing the damage is cut out by a DNA cutting enzyme Nichols, and the resulting gap is filled with nucleotide probably paired with the nucleotides in the undamaged strand. The enzyme involved in filling the gap are a DNA polymers and ligase. DNA repair of this type is called nucleotide excision repair. What's that? One function of the DNA repair enzymes in our skin cells is to repair genetic damage caused by the ultraviolet rays of sunlight. One type of damage, the type shown in that figure, is the covalent linking of thiamine bases that are adjacent on a DNA strand. Such thiamine dimers cause a DNA to buckle and interfere with DNA replication. The importance of repairing this kind of damage is underscored by the disorder xeroderma pigmentum, which in most cases is caused by an inherited defect in a nucleotide excision repair enzyme. In individuals with this disorder are hypersensitive to sunlight. Mutations in their skin cells caused by ultraviolet light are left uncorrected and cause skin cancer. Replicating and ends of DNA molecules, in spite of the major role played by DNA polymers in DNA replication and repair, it turns out that there is a small portion of the cell's DNA that DNA polymers cannot replicate or repair. For linear DNA, such as the DNA of eukaryotic chromosomes, in fact, that the DNA polymers can only add nucleotides to the 3 end and of a pre-existing polynucleotide leads to a problem. The usual replication machinery provides no way to complete the 5 ends of daughter DNA strands, even if an Okazaki fragment can be started with an RNA primer bound to every end of the template strand. Once that primer is removed, it cannot be replaced with DNA because there is no 3 end into which DNA polymers can add DNA nucleotides. As a result, repeated rounds of replication produce shorter and shorter DNA molecules. Prokaryotes do not have this problem because their DNA is circular with no ends. But what about eukaryotes? Eukaryotic chromosomal DNA molecules have nucleotide sequences called telomeres at their ends. Telomeres do not contain genes. Instead, the DNA typically consists of multiple repetitions of one short nucleotide sequence. The repeated unit in human telomeres, for example, is the 6th nucleotide sequence TTAGG. The number of repetitions in a telomere varies from about 100 to 1000. Telomeric DNA protects the organism's genes from being eroded through successive rounds of DNA replication. In addition, telomeric DNA adds specific proteins associated with it, somehow prevent the staggered ends of one daughter molecule from activating the cell system for monitoring DNA damage. The end of DNA molecule that is seen as a double strand break may otherwise trigger signal transduction pathways leading to a cell cycle arrest or cell death. Telomeres do not prevent the shortening of DNA molecules due to successive rounds of replication. They just postpone the erosion of genes near the ends of DNA molecules as shown in this figure, telomeres become shorter during every round of replication, as we would expect. Telomeric DNA does tend to be shorter in dividing somatic cells of older individuals, and the cultured cells that have divided many times. It has been proposed that shortening of telomeres is somehow connected to the aging process of certain tissues, and even to aging of the organisms as a whole. But what about the cells whose genomes persist unchanged from one organism to its offspring over many generations? If the chromosomes of germ cells which give rise to gametes become shorter in every cell cycle, essential genes would eventually be missing from the gametes they produce. However, this does not occur. An enzyme called telomerase catalyze the lengthening of telomeres in eukaryotic germ cells, thus restoring their original length and